Assalamualaikum and a very good afternoon. So I will be taking this lecture from Dr. Nazul Jambari, and these notes is actually coming from her as well. So our topic will be the biochemical changes during fermentation, specifically on glycolysis and lipolysis. So glycolysis is the breaking down of the sugar and lipolysis is the breaking down of the fat. So this is the learning outcomes. You can read it yourself. Okay, just to recap back what we learned in biochemistry during your first year. These are the carbon metabolic pathways. As usual, it will start from the glucose, and the glucose will be broken down into pyruvate. So if you got enough oxygen, especially in a human, because in the microorganism, even if you got the oxygen, you actually or usually don't go into the aerobic respiration, because certain microorganisms they prefer to have no oxygen, although under the presence of oxygen. So if in the human, this pyruvate will enter into TCA cycle in the mitochondria. Okay, they know as you can see in the yellow line here, and the coenzymes in form of NADH and FADH2, this one, okay, they all will go into the next stage of the ATP formation, which is the electron transport chain. This one is happening on the membrane of the inner mitochondrial membrane. And we also learn in the TCA cycle that all the intermediates actually share the same structure as certain amino acid. So some of this intermediate can be converted into certain amino acid. For example, alpha ketoglutarate can be converted into glutamine and also glutamate. Acetyl-CoA can be converted into some kind of amino acid as well. And we also learned that fatty acids can also be derived from the acetyl-CoA. So all the metabolic reaction, they all can meet at some point. They all can converge and they all can diverge at some point. And if you don't have enough oxygen, this pyruvate can be turned into lactic acid. And if in the microorganism, this lactic acid can actually be converted into more products. For example, they can be converted into alcohol or other kind of acids as well. Okay, these are the different kind of fermentation that we will take a look today. Okay, so these are the basic concepts. The first one is metabolism, which is the combination processes of catabolism and anabolism. Catabolism is the processes by which a living organism obtains its energy and raw materials from nutrient, or in short, actually the breaking down of the bigger molecules into a smaller one. Anabolism is the processes by which energy and raw materials are used to build macromolecules and cellular structures. So basically, anabolism is the reverse reaction of catabolism. Means you're building up the molecule instead of breaking it down. Okay, just some more basic concept, which are reduction and oxidation. So reduction and oxidation, they always occurs together in a reduction oxidation reaction. Sometimes we call it as redox reaction. One substances get reduced and another one can get oxidized. The thing that get oxidized is called the electron donor, and the thing that get reduced is called the electron acceptor. So basically, this molecule can give the electron, and we can call that as electron donor, and it can get oxidized. And the one that accepts it, we call as electron acceptor, and it is get reduced. In metabolic pathways, we are often concerned with the oxidation or reduction of the carbon. Okay, in most of the metabolic pathways that we learn in biochemistry, they usually concerned with the reduction and oxidation of carbon. The reduced form of carbon, for example, hydrocarbons, methane, fats, carbohydrate, and alcohols, they all carry a great deal of potential chemical energy stored in their bonds. Meanwhile, the oxidized form of carbon, for example, ketones, aldehydes, carboxylic acid, and carbon dioxide, they carry very little potential chemical energy in their bonds. Okay. For the enzymatic pathway of metabolism, the metabolic reactions take place in a stepwise fashion. It means that they must follow certain steps before they reach into the final molecules. in which the atoms of the raw materials are rearranged, often one at a time until the formation of the final product takes place. Okay, for example, if you are breaking down the glucose, eventually you will produce the pyruvate, which can be the precursor for other reaction as well. Okay, should we take a look in the subsequent slides? Each step requires its own enzyme, okay, specific for each reaction. Okay, you also learn about this one during the enzymology by Professor Nazami. The sequence of the enzymatically catalyzed steps from a starting raw material to final end product is called an enzymatic pathway or metabolic pathway. Cofactors for redox reaction. Just to summarize this slide, the two major cofactors are, the first one is NAD and the second one is FADH, and they always get reduced to NADH and FADH2, which will later on be shuttled to electron transport chain to reform this NAD plus and FAD plus, because these are actually very low. They have limited amount in the cells. They must be recycled in order for this cofactor to recatalyze the reaction again. Okay, that's why we see a lot of this being recycled. Okay, so that other reaction can be done. All right. I think you know the importance of NAD plus and FAD plus during one of the reaction. I forgot the name, whereby the formation of the lactic acid must be transported to the liver, and because the liver needs to turn back this NADH to NAD plus, but I forgot the name. You can take a look later on during the biochemistry lecture. Okay. So for ATP is a currency of energy for many cellular reactions, even in the animal, in the plant, and in all the living organism. So it stands for adenosine triphosphate. It is a nucleotide with three phosphate group linked in a small chain. And if this one is being broken down, it will be changed to ADP, which is diphosphate, means that you only have two phosphate. The last phosphate in the chain can be removed by hydrolysis. The ATP becomes ADP or adenosine diphosphate. This reaction is energetically favorable. It has a delta G of about -7.5 kilocal per mole. Okay, so you learn in the enzymology that in order for the reaction to move forward or favorable towards the product, it must have the negative delta G. Okay, so sometimes in the cell you will see the positive delta G reaction. So in order for you to make sure the reaction can go forward, you can couple them together with highly energetic, negatively energetic reactions such as ATP. Okay, as long as the total reaction will become negative, then the reaction can go forward. Okay. So as you can see from here, the hydrolysis of ATP will produce ADP plus phosphate plus energy, and one ATP is equivalent to 7.5 kilocal or calories. And this ADP can further be broken down into AMP, adenosine monophosphate, right? And they can also be rephosphorylated back into ADP and ATP if we add one phosphate group to the molecule. So ATP hydrolysis is used as an energy source in many biological reactions that require energy. For example, the active transport in the sodium-potassium pump, or even the movement of the human being, or anything that requires energy. During catabolism, the energy released from the oxidation of carbon is captured and used to synthesize ATP from ADP and phosphate. Okay, this is the example of the reaction. Okay, glucose plus oxygen in the aerobic respiration, it will produce carbon dioxide, water, energy, phosphate, and okay, while we have another energy. Alright, this one is actually okay. Confused, these are actually two different reactions. So glucose plus oxygen will produce carbon dioxide H2O and also energy. And likewise, if you want to produce ATP back, ADP plus phosphate plus energy, it becomes ATP plus H2O. For aerobic respiration, it is a process by which a cell uses oxygen to burn molecule and release energy, and also to produce some of the ATP that we can use in the body. So the reaction is glucose plus oxygen. It will produce carbon dioxide and water. Note, this reaction is the opposite of photosynthesis. So for photosynthesis, it's going to be water plus carbon dioxide, and it will produce oxygen plus glucose. This reaction takes place over the major of three major reaction pathways. The first one is glycolysis. The second one is the Krebs cycle, or also known as several names, for example, TCA cycle, and also the TCA, or also the citric acid cycle. And the final process is electron transport chain, or electron transport phosphorylation, because the phosphorylation reaction was happening during the electron transport chain. Okay, another name is chemiosmosis. So for the glycolysis, it's pretty straightforward. Okay, from the glucose, you will undergo several reactions, and it will be broken down into two 3-molecule carbon, and eventually all these 3 carbon molecule can be converted into pyruvate. Okay, so it says here glycolysis produces 4 ATP and 2 NADH, but it will use 2 ATP in the process. In the process we call as investment process, for the net of 2 ATP and 2 NADH. We won't go further into that one. Strictly fermentative bacterium produces energy by glycolysis only, means that this pyruvate will never enter into TCA cycle. They don't even have the TCA cycle or the electron transport chain. Okay, they will produce certain kind of acids, for example, lactic acid, butyric acid, and so on. And also, some of them can produce alcohol as well, as we can see in the yeast fermentation. In biochemistry, for the sake of convenience, the fermentation pathways always start with glucose. So, of course, in the fermentation medium, we can add other carbon sources as well, apart from glucose. But glucose is the simplest one. Okay, but if you add other kind of substrate rather than glucose, it probably can redirect the pathway of the formation of the product into something else. Okay, so instead of producing, for example, lactic acid, if you add for example, fructose, it can probably produce butyric acid instead of lactic acid, for example. Okay, so we're going to use glucose because this is the simplest molecule, requiring the fewest catalytic steps to enter into a pathway of glycolysis and central metabolism, which is the TCA cycle. The 3 major glycolytic pathways found in different microorganisms. The first one we call as EMP pathway. This one is the one that we learn in the basic biochemistry. This is the classic glycolysis, and it is found in almost all organisms, including microorganisms, okay, including yeast and mold as well. The second one, the less common one, is phosphocatalysis pathway, which can be found in certain bacteria such as bifidobacterium and leuconostatic. Okay? And the last one is the ED pathway. It is found in Pseudomonas, Zymomonas, and related genera. Alright. If you see all of this pathway, they all usually will produce the same precursor, which is pyruvate. Only the intermediate is going to be a little bit different. Means that they also use different enzyme to reach the final product of pyruvate. So we're going to take a look at the first one, which is EAMP. This will be continued in the next lecture, okay, because I've already reached my 15 minutes limit. Thank you.